Good morning, everyone. Welcome to another.
Good morning, everyone.
Welcome. Welcome to another exciting day of the high-end workshop, sponsored by Accelerate Vigyan and Department of Science and Technology. Department of Science and Technology, the high-end workshop on computational biology and data science. Today and tomorrow's theme is on drug design and therapeutics. So I have it's an honor to welcome our first speaker of the day, Professor Casey Green from University of Colorado, USA. Professor Green is an experienced leader in the field of data analytics. After completing his PhD in computational genetics in Dartmouth College, 2009, he was a computational biology and functional genomics postdoctoral fellow at Louis Sigler Institute of Integrative Genomics in Princeton University. And in 2014, he was named a Moore Investigator in Data-Driven Discovery by the Gordon and Betty Moore Foundation. He joined Dartmouth faculty that year and moved to University of Pennsylvania School of Medicine. And currently, Professor Green is a director of the Childhood Cancer Data Lab for Alex Lemonade Stand Foundation, Center for Health Artificial Intelligence at the University of Colorado School of Medicine, and Associate Professor at the Department of Systems Pharmacology and Translational Therapeutics at the University of Pennsylvania Perlman School of Medicine, and Professor of Biochemistry and Molecular Genetics at University of Colorado School of Medicine. His lab is part of the University of Colorado system, involved with developing algorithms that integrate publicly available data from multiple data sets to help model and understand complex biological systems. This approach allows investigators to infer the key contextual information required to interpret the data, facilitate the process of asking and answering basic sciences and translational research questions. So it's again, thank you for Professor Green taking his time out and spending a late night with us for this workshop, and I invite him to deliver his lecture. Thank you.
Thank you for having me. This is, I think, my first time giving a talk tomorrow, which is an exciting experience.
Yeah, that's right. It's third there and it's fourth here. So yeah, you are in the
future. Yeah, so I'm really excited to share, some information around how our group really thinks about kind of drug target identification and where we think computational approaches can be helpful. So I had my phone up just to make sure that I was in fact in the correct place and my screen was shared correctly, and it looks like it's working. So let me close that so that I'll have enough bandwidth to.
Oh, okay. I thought that was another doppelganger. So okay, now it makes sense.
No, I was just making sure my screen share worked.
Okay.
And yeah, so I really recently just moved to Colorado. It's really nice here and truly remarkable, but really fun to be. The excitement and enthusiasm at this campus is really wonderful. So I can speak highly of it for my first month or so living here. Let's see. So, this is, the way kind of my lab approaches really thinking about these types of problems. And this is a letter that Vannevar Bush wrote to the US President Franklin Delano Roosevelt after when he was really trying to make the case for a national basic science effort in our country. And he really taps into kind of this the sort of mentality of exploring the unexplored. So he says, this pioneer spirit is still vigorous within this nation. Science offers a largely unexplored hinterland for the pioneer who has the tools for his or her task. And I think this is, really the way we approach this problem. And, for us, this is really data, like the large collection of data that exists. Many of these data sets have been analyzed, one or two times. And beyond that, they haven't really been sort of deeply delved into, and they also really haven't been analyzed together. And so we think there's a real opportunity here to, really go out into the unknown and come back with some things that are really important and valuable. Oh, and I should say, if you have questions, please stop me as we go, and feel free to ask. That would be much appreciated to ask as we go along. So the first thing, where we kind of started thinking in this avenue is really trying to understand what types of evidence support existing drug treatments. And as we were thinking about this, there was actually this open science project going on at the same time. There was this student at UCSF whose name was Daniel Himmelstein, who was really working on this large-scale systematic integration of biomedical knowledge into a knowledge graph. And now sort of we think about knowledge graphs all the time. This is a thing that's becoming much more widespread. But at the time, I thought this was a really unique and interesting resource. And actually, Daniel ended up joining my lab later and doing a postdoc in my group, which was really exciting. But I thought this was a really interesting tool to answer a whole bunch of questions around how we can really understand what data resources are actually valuable for understanding how drugs work, and also for understanding how we might develop new ones. So the Hewcbds knowledge graph has, just to give you a brief introduction to the graph, it has some entities in it, which are of various types. So, one of the node types is actually pathways. And then it also has another node type of genes. It's a heterogeneous knowledge graph. So genes participate in pathways. Genes also participate in biological processes. Genes can co-vary with each other. So they can sort of, in this case, it's gene expression. So they can co-vary. They can have similar gene expression profiles to each other. They can physically interact with each other. They can be upregulated in certain tissues. They can be downregulated in certain tissues. They can be expressed in certain tissues. So this knowledge graph really tries to capture all of these types of relationships. And these relationships were chosen in particular because they were relationships that could potentially make sense for understanding whether or not a compound would treat a disease. So the idea would be, this is what we really want to know, but we know very few cases of where compounds treat disease. But perhaps we can reason over these other types of edges and understand, what other compounds might treat a disease in question, or what diseases a compound might treat. And I thought this was a really compelling data resource that Daniel developed. And this is actually from Daniel's papers before he joined the group. What he did is he trained a classifier to predict treatments. So what you'd imagine is you have a set of treatments and non-treatments. And then you have, for each of these features, you can give a weight to it, which is essentially sort of how much evidence that feature is providing. And then you can simply ask, if you have these treatments and non-treatments, are there certain types of relationships or certain paths through this heterogeneous knowledge graph that predict a treatment versus a non-treatment? And then the idea is, if there are, then you can find other paths, other compound disease relationships where those also exist, and predict that those would potentially be treatments. This actually works pretty well. There's a few different ways you could evaluate it. So the first one is you could ask, how well are disease-modifying treatments prioritized by this type of approach? And you actually do a really good job of predicting disease-modifying treatments. So this is an AU-ROC plot. The perfect AU-ROC plot is one where it goes entirely up before it goes over. And you can see the disease-modifying treatments are successfully prioritized. The other thing you can ask is sort of, are treatments that go into clinical trials prioritized? It's also able to prioritize sort of what is going to be put into clinical trials. So this also, you could say, also probably recapitulates some of how people think about pharmacology, which is nice. And then, this to me was the real kicker that was very eye-opening, because there's a lot of different ways you can imagine evidence accruing when you have a drug and you have a disease. And so what this is showing is it's broken each of those long paths. So here, we might have some really long paths. So this has compound binds a gene, which is expressed in an anatomy, which is implicated in the disease. So compound binds gene expressed in anatomy, implicated in disease. And so, or local disease that the disease localizes to, actually, that's an L. And so then what we can do is, so we have disease localizes anatomy. So this is one of those edges. So that this edge here, this edge type, is one of these dots on disease localizes anatomy. And so each of these paths, what we can essentially then say is, if you have a path that includes the edge compound treats disease, how informative is that? And for each path, we can put a dot on it. And you can see that compound treats disease is among the highest value edges and how this usually works, the other ones here that are high value, are that a pharmacologic class includes a compound that treats a disease. So if you put these two together, you can have a length 2 path where a pharmacologic class includes a compound with another compound that treats the disease. And that's actually one of the highest value edges in the entire data set. So, that's kind of how this starts to work. And so what you can see here is these really break out nicely into sort of so traditional pharmacology, we really think about, what compounds treat disease. We think about pharmacologic classes. We think about similarity of compounds. We think about similarity of diseases. And we think about direct targets, what gene products are bound by compounds. So, this suggests to us that traditional pharmacology is really pretty high value. This is really kind of genetic association based. So, this is kind of the GWAS, disease associates gene. And there's also some other sort of things in here that are now being gene-gene interactions are all being used for kind of drug efficacy. So you can see that these also have sort of a high value, not quite as high as what we see in traditional pharmacology, but still have some value here. And then this is kind of interesting. So, all of these are a little bit targeted. By and large, they're not genome-wide relationships. And they're sort of generally reasonably biased data sources in that the only way something shows up in those data sources is that someone chose to look at it. As we get down to the genome-wide data, you can see that there's still value there. Certainly, these are generally to the right of 0, which would be sort of the point at which you're not really getting any additional value, but they're a little bit lower. So on the individual case, even though in aggregate, they're still quite valuable because they're genome-wide. So I thought this was really interesting as a way of tackling, what is the real value of understanding different types of relationships. I'll come back to this later, but this is actually still dependent on the methods that you use to evaluate these. So I think there's ways to actually make these more valuable, and we'll talk about that later. But just for the moment, knowing that, the sort of base way that you would use these, there is value there. They're just a little bit more limited in what they can provide. When Daniel joined the group, what we wanted to do was expand this a little bit. And so the previous work all used supervised machine learning at some stage to weigh the strength of the evidence. And what we really wanted to do was to be able to do the same thing without having to do supervised machine learning, so that we could expand this not just between a compound and a disease, but between any pair of relationships, gene disease, compound to tissue. And that was really sort of the inspiration for this project. And so this is Ptolemy, translated, but this is essentially also sometimes phrased as Occam's razor. We consider it a good principle to explain the phenomena by the simplest hypothesis possible. So we wanted to do the exact same thing over Hewcbds, where I slightly reframed it to, we consider it a good principle to explain the relationships between biomedical nodes by the simplest corrected hypotheses possible. It turns out to be very easy to do shortest path, so the simplest, but short paths are often generic. And so correcting for that generic nature turns out to be really, really important. I know y'all talked about networks pretty recently. And so I suspect some of this came up, and a little bit of this may be review. So one of the things that's important to think about is that, you probably have talked about or may have become aware that networks, different nodes in networks have different degrees. So some nodes are highly connected to many other nodes, and other nodes have relatively sparse connectivity. And there's very different patterns of connectivity that can exist in different networks. And this is what we see here. So the compound resembles compound. So these edges often have relatively low node degree, relative to kind of the overall network. So these are sort of relatively tight, kind of hubby types of edges, but very small groups, and a kind of similar size. Here, on the other hand, the anatomy expresses a gene. So there are some anatomies that have thousands of genes expressed in them, and then other anatomies that are pretty specific. And so if you don't account for both the node degree and the distribution, the different distributions, life here gets pretty challenging. And actually, this was a little bit surprising to me, and is worth noting. So for certain edge types, we have both biased and unbiased edges. So the way these are labeled here is systematic versus literature derived. And so literature derived or biased, right? That means someone went in and did an experiment to try to understand that. Systematic is from a genome-wide screen. And the distributions between these two are actually also different, which suggests that, you could imagine that these node degrees are really real and, just like it is what it is. And some things sort of have this degree distribution and we can get a lot more information out of them. But actually, because so much of the network is derived from curated literature and less is systematic, seeing that there's a big difference between the literature derived and the systematic suggests that we really do need to correct for this. And I'll just note that this is seems to be more a property of sort of biological networks derived from like these types of experiments. So these are all kind of standard biological network types: protein-protein interaction, transcription factor, target gene relationships. When you look at co-authorship in bioarchives, so this was just another type of network we could generate, we don't see this same relationship, when you have a year cutoff. So it seems like there's really something fundamentally different about these types of networks and experiments. And this is just showing that if you try to use the systematic experiments as a holdout for the literature derived, you actually don't predict very well. Whereas if you do sort of just a subsample, obviously that's pretty predictive. And so this really suggests that you need to think about correcting this. If you just do shortest path, you're probably going to be really heavily driven by experimental bias and curation bias. So we put this together, ended up, there's a whole bunch of sort of methodological work that goes into it, but now there's actually a tool. So if you're interested in this, you can play around with it. We're pretty excited about it. We think there's a lot of opportunity here to think about using this as sort of a first pass filter for when, if you've discovered an association, you might ask, what are the potential mechanisms by which that might actually be working? And so if you want to play around with this, go to the search.het.io website. You can kind of follow along with the examples we'll go through. And if you want to bookmark it, you can come back to it and keep using it. But the way this works is sort of the many possible ways you could connect entities, which ones are incompatible with simply knowing the node degree in the network structure, or for which ones is there sort of some apparent biological support that goes above and beyond just sort of these distribution of edges. So you can use this for a whole bunch of things. So this is asking, which diseases are most related to a molecular function. So we put in a molecular function here, IL-23 receptor binding, which we were potentially interested in for a certain type of project focused on a specific disease. And first, one of the nice controls is that the interleukin-23 receptor binding, the most significantly associated connection to that is the interleukin-23 receptor as a gene. So that's promising. And then you can also do something where you can filter, you can deselect these nodes at the top and only pick one node type. So you could say what diseases, this is asking what diseases are most related to IL-23 binding. And you can see there's a lot of autoimmune diseases here, which makes a lot of sense. And was sort of one of the things we were really interested in with this project. This paper we're still writing, but please feel free to play around with this website. We've been sort of diving a bit deeper back into the drug development sort of target identification approach with it. And we're pretty excited about where that is taking us. There's 3 new software packages. So what I've told you about is the web server where you can run it yourself. If you want to do it at scale, there's 3 different software packages here that are tied together to really make that web server work. So if you wanted to implement this on a genome-wide basis yourself or over your own heterogeneous knowledge graph, this is the tool that would let you do that. There's actually 2 papers behind this. One of them is up on GitHub. We still need to finish it up and submit it to a journal. This is the one led by Michael Zietz, the probability of edge existence due to node degree, which really looks at this sort of degree distribution challenge. And then the next one is Daniel Himmelstein's paper on search over heterogeneous networks, which still lacks a full author list and a title, but is now going up on GitHub. And so, this is really what got us thinking about how these high throughput data sources can be used for drug development. And, one of the things I want to go back to in this from Daniel's work that I thought was really interesting is that, this sort of gene regulation information and sort of gene co-expression, gene pathway information, it seems informative, but it does have some limitations, right? The benefit is that it's genome-wide. You can get predictions for every gene. But the challenge is that, the way we're currently using the data and sort of the most straightforward approach, it's really not that informative on a individual basis, even though globally it's pretty informative. And the other one that we really care a lot about is sort of gene covariates with genes, so which genes are co-expressed together. And there's only a little bit of signal here in the way we usually approach this problem. So we started to ask, is there a way we can get more value out of these gene expression data? Actually, let me pause here. Does anyone want to ask a question before I switch? We're going to go pretty deep into gene expression next, but this was kind of the survey of types of evidence.
Hi, Casey. This is Pulkit. So just wanted to ask why we are, focusing in depth for the gene expression rather than, combining all the multi-omics layers, let's say from starting from variants, epigenomics, and then gene expression, obviously, and then going to protein expression dataset and then metabolites expression dataset.
Yeah. So I mean, I think that's a really interesting question. I think the answer is we have to do both. There's a lot more. There's a lot of limitations on sort of where you can sort of get multi-omic profiling, especially of sort of key high-value samples. I think in the long term, there's going to be significant benefits from combining across different omic data sets. I will say, we have a project in the lab where we've sort of asked, if you're going to think about sort of functional consequences of, say, somatic variants, what omic layers can you best detect them in? And actually, steady state mRNA is a pretty darn good predictor, which was surprising to me. Even for things where you wouldn't necessarily, like, there were certain types of mutations where I would expect them to have more influence on a different omic layer. And I guess what I would say is I'm coming around to the idea that it matters less which omic layer you profile, except in some very specific circumstances, because the hallmarks of perturbations, while they may be different on different omic layers, are likely to still exist in some fashion. But I do think for key processes, going kind of deep through the multi-omic profiling is going to be helpful. But I think from a broad survey point of view, I used to think, you needed both, but I'm starting to come around to the idea that it's okay to sort of pick an ohm and use that for very wide integration, and then sort of take some key high value samples for your problem and then profile them very deeply. And I think those two might actually be combinable. But yeah, it's a great question, and definitely kind of an area where my thinking is continuously evolving.
Thank you. And just one more question. So I have used head.io on Neo4j, and it was, a really great tool. What I could think of means if I'm integrating, let's say, variant data into it or miRNA data, so would it be adding any value? Means many of the diseases occur just due to mutations in our genomes. So just
Yeah.
thinking on that direction.
Yeah, I mean, I'm talking about Daniel's resource and he's not here. So I'll one of the things I'll say is, the gene associations with disease, particularly if you're interested in malignancy, they're all based on sort of GWAS genetic associations at this point. And I think there's some significant opportunities to improve that network by including, things like somatic variants and the consequences of somatic variants. I think that would really help in many projects. Adding additional data types. I think what I would expect is, the additional data types are probably going to largely end up in this case, like in this region, but that's okay, right? Because if they're genome wide and they're just adding a bit more evidence, that's totally fine and it's really valuable. So I guess I would encourage that. Daniel maintains Hedio, and I think I don't know how much it is still getting updated, and I know that he has some pretty rigorous beliefs on what the licensing sources should be. So I don't know if that should get fed upstream into Hedeo or if that should get added in like as a layer downstream, and then you could launch your own instance that includes it. If you're interested, I would definitely go to the GitHub Hedeo. It's actually an organization, and maybe on HedioNet, file a GitHub ticket and see what he thinks. Hopefully that helps.
Yeah. Thank you.
Any other questions before I wander onwards? Okay, I will wander on. I will say that the methods that I'm about to present and talk about, I think are likely, there are almost certainly equivalent type approaches that you could develop for the other omic layers. We haven't, by and large, done that yet, although we're starting to explore some, especially kind of epigenomic modeling with the similar types of approaches. Okay, so our hope is really some new methods can help us push these up, and I agree. I think adding other ohms can also potentially push this up. This project, is really trying to understand how we can think about sort of gene modules, and you could imagine the output of this being gene modules that then get fed back into Hedeo as a new type of relationship, and we might hope they'd push things up. We have not done that analysis yet, but it's sort of something we've been thinking about. Okay, but in this case, we really wanted to understand, if you have a few different data sets from a disease, what's the best way that you can, what's a good way to analyze them across data sets? Because if you have tried this before, I know you had an RNA-seq lecture earlier. If you've tried this before, it turns out to be pretty hard. Analyzing individual data sets we're still okay at, but like looking for consistency across tissues is pretty tough. And you might think, well, I'm probably not going to generate a whole bunch of data sets, but it's worth noting that this is a back of the envelope calculation that there's about 4 million gene expression samples out there, which is about a $4 billion resource. So I suspect that if there's a data set that you have, there's at least 2 or 3 other data sets that exist somewhere that are publicly available that are potentially valuable for your scientific question. And I think analyzing your data in the context of other data sets is likely to reveal more than sort of analyzing individual data sets alone. So those of you who have gone down this path know this actually turns out to be pretty darn hard. You would hope it would be easy, right? The idea is conceptually simple. You analyze a data set, you find some sort of modules that are interesting in that data set, you analyze a different data set, you find some of those modules, you analyze a different data set, you find some of those modules, and then all you have to do is see which modules are consistent across the data sets. And the real challenge with this type of approach is because we usually look within data sets, the modules are often vastly different because there's data set specific technical artifacts. Sometimes these data sets come from different tissues. And so saying, is this module really the same as that module is actually a pretty hard problem, and it requires some deep domain expertise. So this is a project led by Dr. Jacqueline Tironi, who was a postdoc in the group and now works with Alex's Lemonade Stand Foundation as the head of the Childhood Cancer Data Lab, now that I am in the process of moving on from that role. And what Dr. Tironi said was, man, wouldn't it be really nice if you had this library of modules, gene expression modules that were reusable, and you could take those modules, and instead of having to learn the modules within every data set, you could just learn the modules once and check in each data set, because then every one of your modules has an index, and you can just check to see if that module is doing the same thing across your data sets. And so this was her idea and approach. And we were really interested in rare diseases in particular, but this approach should be applicable not just for rare diseases, but to any high value data set. And when you're thinking about drug development, one of the things that is becoming common is to sort of do omic profiling of samples from that data set to look at potential sort of biomarkers of response or non-response. And I think this type of approach is really well suited for that type of challenge. So in what you'd really love to have to run this is you'd like to know sort of some things about a whole bunch of people, and then you'd like to use that to build those modules. For rare disease data sets like the ones we were looking at, or if you're thinking about kind of clinical trials data sets, you have this challenge where you can sort of do genome-wide profiling, right? You can measure the expression levels of 20,000 genes. You can measure the expression levels of thousands of proteins. You can measure the expression levels of, hundreds of thousands of methylation marks. But at the end of the day, you're only going to have these measurements on a few people. And so somehow, you have to make this work. Jacqueline's approach was to say, well, what if we didn't make machine learning models for just our data sets and just our contexts? What if we made it for many contexts? And so, to state the hypothesis a little bit more formally, her hypothesis was that these biologically meaningful patterns, you could learn them from heterogeneous gene expression data and then transfer them to the data set of interest. So if you had sort of samples by genes, you could take these and you could learn these patterns on this sort of generic gene expression data that sort of wasn't specific to your process of interest. But then you could take those patterns and look for them in your rare disease, or look for them in responders and non-responders in your clinical trial. And you could say, are there patterns that are associated with either disease or control status? Are there patterns that are associated with response? And this was really, I thought, a really interesting insight. So we decided to pilot this on a data set of 70,000 RNA-seq samples. If you're interested in sort of generic human data, and you want 70,000 RNA-seq samples, but you don't want to spend the CPU hours to process them yourself, I can highly recommend Recount2, which is from a group at Johns Hopkins. And it's from Jeff Leake's group at Johns Hopkins. And so they process 70,000 RNA-seq samples. If you just want to benchmark out what this data set would have cost to generate, if you imagine on average that costs $1,000 a sample, it's a $70 million data set, which is not a negligible sort of resource investment, to say the least. We're going to apply a method to that. So the method, we tried a few different methods, and the one that we found performed well for this problem was something called Plier, the pathway level information extractor. It's from Maria Shakina's group. So Maria has a lab at Pitt, and Wayne Mao is the first author who developed this. The way this works, you're essentially taking this really large matrix and you're going to break it up into patterns. And you're going to do that in a way that tries to, there's some sparsity constraints. So each of those patterns is going to have some genes that have a whole bunch of, some genes that are 0, and then some genes that have weights to them. So it's going to try to make genes have, at least a reasonable number of genes have 0 weight. So you don't have to think about them. You can just think about the pattern as the set of genes that have weight. And it's also got this nice thing that it does where if a latent variable or if one of these sort of patterns kind of looks like a pathway, the method gets a little bit of a boost from adding more genes from that pathway. And so what that means is functionally it doesn't dramatically affect the fitting, but it really makes this model more interpretable because each of these sort of patterns, if it can look like a pathway, sort of does look like a pathway. So it makes it really aids in the interpretability more than the model fitting. And we're really happy with that. It really makes it nice to reason over the results. And so so if you take multi-data set Recount2, add it to Plier, it gave us something that we call multi-data set Plier, or multiplier for short. You might ask, well, maybe I should have just used data from my specific disease. Maybe I didn't need Recount2, which is this large collection of generic human data. So we wanted to ask that question and see if sort of getting outside of the disease context was helpful. We looked at, we were interested in autoimmune diseases for this. So we actually, the autoimmune disease we actually wanted to study was ANCA-associated vasculitis. It turns out there was not enough data for ANCA-associated vasculitis to actually really succeed at building these models. And so we had to find a different disease that we could sort of use for this at least evaluation. So we looked at lupus, so SLE, and we downloaded all the whole blood data for lupus, and that gave us one data set. And then we used Recount2 as our other data set. We also did an experiment here where we sort of subsampled Recount2 to be the same size as the lupus collection. So where you see these box plots, those data sets are the same size, but different composition. This is lupus whole blood. This is generic human data downloaded from the internet. Then the other thing we did is we compared, so this is Recount2 subsampled. This is the entire collection of Recount2. And so where you see this diamond, it's going to be the same type of data. It's just a vastly different number of samples. And so the first thing we can ask is kind of how many things do we learn? So you get more total latent variables. You can learn more patterns, reproducible patterns, if you have more data. So that's not terribly surprising. The other thing that's not terribly surprising, but is a little bit interesting, if you use generic human data from the internet and you match the sample size, you actually get fewer patterns than if you had the disease-specific data. So like, if you have a limited amount of data, you probably do want to sort of, if you have a limited sample size, you would like your samples to be relevant. But on the other hand, because you're willing to relax that relevancy constraint, you can get a lot more reproducible patterns if you have more data. So you can learn kind of more total things. The way this method worked, because you sort of have that ability to sort of align these to pathways, you can ask how many of the things that come back are aligned to pathways? Like what proportion of the pathways that I put in did I get some transcriptional pattern back for? And we don't know where this tops out, right? This question came up earlier, but what ohm do you choose, right? Some things are not transcriptomically co-regulated. So this, we would not expect 100% of pathways to have some transcriptomic co-regulation. So we don't know what the top of this plot should really be. But what we do know is, it should go up, right? The more we can detect in the transcriptome, probably the better. And so what you see here is there's actually not that big of a difference by dataset composition, but the size is a really nice driver of sort of being able to recover more transcriptional signatures that align to pathways. And this goes to the what ohm question. If you did integrate multiple ohms, if you had enough data to sort of get multiple ohms, you'd really expect this would sort of start to increase, because you'd be able to get things that aren't transcriptionally co-regulated, but maybe have sort of coherence at the protein level, for instance, or even metabolite level. So you kind of learn more total known knowns. And then the other thing is, are you learning anything new here? Or are we just recapitulating the pathways we put in? You can see here about half of the latent variables that come back in the sort of smaller data set models are sort of already things we know, right? They're sort of pathways that are well annotated. So you could think of these as things you might identify with GSEA or pathway overrepresentation analysis. But when you get to these really large, the models built with really large collections of data, like the full recount2 set, only about 20% of those patterns that came back were things that we sort of have a priori labels for. So 80% of the things that come back would have been transcriptomic patterns that you would be blind to if you were only using curated databases and not the sort of data derived latent variables as well. So we thought this was kind of exciting and a nice way to understand what more data really get you in this context. So there's a whole bunch of stuff in the paper. I've really just sliced out like 2 or 3 figures from the paper. But this paper is sort of one of the densest things that we've written in the last few years. And so I would really encourage, if this is of interest to you, sort of, there's a lot more in the paper. The take-home message for us has been that these machine learning analyses that reuse data can actually reach a level of detail that's impossible if you're only analyzing data from the specific context that you really want to understand. So just to remind you, the challenge with the modular framework was you derive these, then you have to match them up. That's really hard. The nice thing about multiplier is you can take them, you can transfer the model, and then you can look across. So now we'll do this analysis. So this is a multi-tissue comparison. So this is a type of thing that is just brutal if you have to sort of compare, because a certain pathway in the lung may look or in the airway epithelial cells may look different than the kidneys. So this is renal glomeruli, which may look different in blood. So the benefit of multiplier is now we can say, are there transcriptomic signatures that are consistent across these 3 data sets? And we can just ask that question really simply by looking at latent variables and asking if there are any that are consistent. So one of the strong signals that's consistent across these data sets is that M0 macrophages are elevated in severe disease. So each of these data sets is very different. They have different composition, they're different tissues, but the key is that we've ordered them from the most severe form of the disease on the left to the least severe form of the disease on the right, or in this case, healthy controls. And what you can see is the severity is nicely associated with sort of M0 macrophages across each of these. So you might think of M0 macrophage signature as sort of indicating a kind of systemic response to, that's associated with disease severity. So you might think about whether or not you could tamp down that M0 macrophage signature if you wanted to try to approach some symptom mitigation for this ANCA-associated vasculitis. A couple things that are interesting on this. I don't want to go too deep into the technical weeds on it. But this is actually, each of these data sets is actually a microarray data set. There weren't a lot of RNA-seq data sets available to us for this disease. And so this model trained on RNA-seq data actually works pretty well on array data, which is a nice feature of it. The other thing that was interesting is there's actually no ANCA-associated vasculitis data at all in RECOUNT-2. And so, this model is really learning these signatures from data that are not disease-specific, and then we were able to get some value out of applying them in a disease-specific way. There's a lot of GitHub notebooks for this. In particular, the things that I wanted to highlight were, there's a notebook here looking at predicting rituximab response in this disease. And so this is actually a clinical trial looking for responders, non-responders. And so there's a lot of stuff. I would say the first half of this probably made it into the paper. The next quarter of this made it into the supplement of the paper. And the final quarter was just, stuff that we did and thought was really interesting, but didn't have room to discuss in the paper. So there's quite a lot in this GitHub repository if you're interested. And so, yeah, and the nice thing about this, the other nice thing is this is pretty reusable. So there's now at least one other paper that has taken these and reused them in a completely different context to study a different rare disease. And so our hope would be that sort of, this would be a resource that could be integrated back into, a knowledge base like Hetio, and then we could evaluate, what this tells us about disease, drug disease pairs, for instance. And we haven't done that experiment yet, but, it's definitely something we're really interested in. And what you might imagine is, maybe gene covaries disease is somewhere, but maybe gene shares latent variable with gene covaries gene is somewhere, but we'd hope that gene latent variable gene is somewhere higher up, right? So somehow there's more information in that. So I usually get asked at this stage in these presentations, wait a sec, you just took a whole bunch of random data sets and put them together. How does any of this work? This is generally the question, because often people have thought about batch effects and technical artifacts, and we would just think batch effects and technical artifacts would just utterly confound the signal, and so we'd have no hope of any of this actually working. And there's a lot of papers on this actually, so it's interesting. There's a whole body of literature that argues that sort of these technical artifacts are going to destroy the signal, written by people I respect a lot, including, we use Jeff Leake's data for this project, but Jeff Leake has a paper that sort of talks about the widespread and critical impact of batch effects and high throughput data. And so, I think there's a lot going on here that sort of argues that technical artifacts are going to destroy any chance this has of working. We also have a whole bunch of papers, including some from our group and some from other groups, that argue that actually biology exists in these settings and can be found. And so like reconciling this has been a real area of interest for our group. And so this is a project led by Alex Lee, who's a PhD student in the group, who really tried to delve into, okay, how does any of this literature make sense together? And as we're thinking about drug development, can we really be using these types of large scale approaches, or do we need to think in this space? And so what Alex has done, this is slightly technical, but I'll try to give you the intuition behind it. So this is a certain type of neural network. And what she's done is she's taken a neural network where she can take an input data set, she trains a neural network to reduce it to some smaller dimensional space, and then she trains a neural network to then give you the original data set back. So this is what makes it an autoencoder, is that the training objective is simply to make the new data look like the original data. And the other thing is that reduced dimensional space actually follows some certain distribution. The neural network is trained to follow a certain distribution, which means that then you can actually sample from that space and simulate entirely new data that have never existed before, but that look like genome-wide gene expression data. This is kind of an interesting technology. Certainly something to be aware of and think about, especially from kind of a forensic bioinformatics point of view. Like any technology, there's potential benefits. There's a lot of experiments that you could only do if you could simulate genome-wide data, really thought experiments. Also a lot of potential hazards. We would certainly not want our genomic databases to be filled up with simulated data. There's certainly a track record of people faking Western blots or other types of sort of imaging results, and we would not like to see that in genomics. I think these data sets actually have some characteristic features that would make them quite detectable automatically, at least at this stage. So I'm a little bit less concerned about that here, but definitely a thing to be aware of, especially as you're starting to look at papers of this type of thing. Is you can now simulate genome-wide data reasonably effectively. So this is just taking one compendium, Pseudomonas aeruginosa. So she's downloaded the data for Pseudomonas aeruginosa, basically the same thing as before, essentially everything that's publicly available. That's shown in the white dots. She then uses her VAE to simulate data. You can see that the blue dots are not terribly different than the white dots. There's a little bit of variance compression here, which is how you can kind of, I think, detect that you're looking at simulated data. If someone was trying to be nefarious and pass simulated data off as real data. I think it's reasonable enough to start using for thought experiments. And then what Alex does is she adds technical noise to it. So she takes this, she simulates data in one full model. She can then she can take it, she can partition it out, she can add technical noise to those partitions, stick it back together, and then she can ask how similar these two things look. So your original sort of simulated data and your new data, because these should look, the question is, do they look the same fundamentally? Yeah, so this is just showing, she's also got a procedure that's based on this technique called latent space translation, which doesn't really matter exactly how it works, but just it's worth knowing that these data sets look pretty darn similar to real data. So one of these is actually a real data set. One of these is a data set that's produced through the synthetic method. The way this works, you can have a template data set, you can produce a new data set from a template, and you can just see they look pretty darn similar. I'm the type of nefarious person who, if you work with me, I will occasionally want to know if our methods are working. So I sent a message to one of our collaborators in Deb Hogan's lab to see if they could guess what the experiment was from simply the differentially expressed genes. And they came up with a pretty nice summary. It's pretty cool for me because this is a synthetic data set. It's nice to know that you can come up with a biological story around an entirely synthetic data set. This is the punchline of this paper. So I wanted to skip to the punchline in the interest of time. If you think about technical artifacts and you try to correct for them, as you have a small number of distinct groups of data that have technical artifacts in them, you're much better off correcting, right? As you correct, you only have to take a little bit of a penalty of, this is a similarity metric. So you're only looking a little bit less similar as you correct. However, without correction, if you have a sort of small number of different partitions, your data look really quite different from your original data. And I should add, this dotted line here is permuted, just randomly permuted data on both of these plots. So you can see, we're putting a lot of noise in these data, and if you don't correct, it basically destroys the signal if you have a small number of divisions of the data. But when you get out here where you've got hundreds of experiments or hundreds of different sets of technical artifacts together, now all of a sudden you're actually better off not correcting than you are correcting. You're continuing to take this hit every time you correct your data. And if you didn't correct in the first place, you're actually better off. And I think this is interesting. So this gives us an idea of how these two bodies of literature can coexist, right? If you imagine a line drawn through here, when you're on the left side of that line, batch effects and technical artifacts are pervasive and they will destroy any signal. But as soon as you get out to the right side of that line, you're actually better off not correcting and just sort of going with it. And so I think that's why, if you look at those papers that have been published, the ones that sort of are looking at hundreds of data sets at a time, and not having to do batch effect correction or technical artifact correction, they're sort of looking at hundreds of data sets. The others are looking at tens of data sets. And I would say this simulation study and where this crossover point occurs is going to be really dependent on your assumptions. So I would not take any specific cutoff as a grain of salt, but I was really happy that we were able to at least resolve conceptually how these two bodies of literature can coexist. And so here's the take-home message: these compendia-wide analyses can work because the underlying biological signal is consistent while the technical noise is often experiment specific. So this was kind of a fun thought experiment. I should say this just got published pretty recently at the end of last year in GigaScience, and there's a GitHub associated with it if you want to go simulate your own gene expression compendia. So, I think from a drug development standpoint, this is really important to think about, because the types of analyses that you would do are fundamentally different if you think you're sort of very limited and you have to correct for artifacts, versus if you can sort of think across very large collections of data. And so I think the way to go is to really sort of think about how you can use very large collections of data to sort of generate the ingredients for the analysis that you want to do, whether that's gene modules, whether that's correlation structure. And then think about how you can take that and use that to amplify the power to analyze your really valuable target data set, which is what you usually get when you're sort of looking at early stage clinical trials and trying to understand if there are biomarkers of response and these other elements. And so I think this is one of the ways that really gene expression data can sort of move its way up in the hierarchy of how valuable different evidence sources are. And not just gene expression data, but kind of other omic data. I suspect the same. It would very much surprise me if this wasn't general to other types of omic data as well. And then in the last couple minutes, I just want to share one really quick vignette that I'm having a lot of fun with. And I think, the other thing that's worth noting as you're sort of looking at data from clinical trials or other perturbation experiments is that there are certain genome-wide results that everybody finds. And so this is another story that Alex has led, that she is pushing forward. And and then, this is the thing I usually get as soon as I say that. Wait, there are results that most experiments share. So like, just generically, there are things you're going to find if you do a perturbation experiment. And the answer turns out to be yes. So there's a couple of papers on this. Actually, one of the earliest ones is Gash et al. From the year 2000, but it's in yeast. And it sort of, I thought it was a really interesting paper, but has not, it was sort of pitched as a generic stress response and kind of was acknowledged in yeast, but hasn't been deeply followed up on in other contexts that I'm aware of. But there's been a couple papers over the last few years. So this is one actually out of the University of Colorado, which is where I now am, but from before my time there. So this is one led by a student, Ronnie Powers, senior author Jim Costello, that created a method they called GSEA in context, which was trying to filter out these kind of generic pathway findings, because there are also certain pathways that come up over and over again. And this is a paper from Jesse Gillis's lab at Cold Spring Harbor, led by postdoc Megan Crow, that argues that differential gene expression is actually predictable. So you don't even have to do the experiment. You can know what's going to be differentially expressed, which is kind of provocative argument. And both of these are really based on the same underlying idea that there's some genes that are just generically differentially expressed. And so, you might imagine, well, okay, I'm going to do my analysis of my, perturbation experiment or my clinical trial, and I'm just going to filter out the generic stuff because I want to know what's specifically changing when a person is exposed to this drug, or when I put this drug on cell culture. Well, why don't you just filter these, right? All you have to do is have 1,000 equivalent studies, and you go through those 1,000 equivalent studies and you analyze those, and you just filter things that come out across, say, 50% of those. Well, the reason you don't do it is because you have to curate those 1,000 additional studies. Switching contexts, if you want to change your methods, any of that requires you to recurate and reanalyze all of this data. So it's really time consuming and painful. But it turns out if you can simulate entirely new experiments, then you can simulate generic experiments. So you don't actually have to do the other experiments. You're going to simulate a null background. So the way this works is, again, Alex is using that same simulation technology from before, but now instead of sort of looking at sort of data set similarity, she's calculating differential expression. And then she's ranking genes by the differential expression in the simulated data. The nice thing about this is, all you need is unlabeled gene expression data to train the model, so you don't have to curate. You can use it for arbitrary platforms, organisms, and this goes to the multi-omic question. You can use it for other omes, and you can plug it into any analytical strategy that you could use with that experimental design or with that experiment, because you're basically simulating data that match a template. So you just have to push it forward through the same exact analysis process. So it's much, much easier to plug into your workflows. And so this this is looking at the Powers et al. Data using the simulated method. So this is microarray. So this is Powers et al. Was microarray data. This is the differentially expressed prior data set or rankings that they provided. And so this is a heat map where white is showing an increased density, black is a decreased. density. And you can see the most generic genes and the least generic genes kind of have some commonality here. So this is microarray, and these are also both curated data sets. So you could have just done this without this method. On the other hand, we're going to take our favorite RNA-seq data set, this recount2 data set, and you can now do the same thing. So this is actually doing RNA-seq data's generic differential expression, and this is doing microarrays differential expression. And you can see that these also have this kind of nice pattern where things that are generic in the microarray, things that are generic in the microarray are also often generic in the RNA-seq, and things that are sort of not generic in the microarray are not generic in the RNA-seq. And then you've got this funky little cluster down here of things that are only observed as generic in the RNA-seq. Have two potential explanations for this that we're actually still digging into. And then, so yeah, I don't have an answer for what those are, but I'm interested in that question. And then another thing that I think is worth noting, I mentioned before that pathways are also generically differentially expressed, so you can plug in your pathway analysis method. This is actually looking at the powers at all ranking versus the simulated method. And you can see you get this kind of nice positive correlation here too, where things that are generic in one are also generic in the other. I didn't put this slide in, but you can also, because you can do this over any method, you can actually compare methods to each other. And things that are generic in one method are also often generic in another method. Yeah, so you can use this with arbitrary gene set enrichment methods. So, I think this is kind of interesting. Whenever we do these perturbations, we will often see some common pathways. And we like to think, oh, this is a real interesting connection between this and that process. But it's really an interesting connection between this and essentially any process. And so, maybe we should focus a little bit less on the hyper-responsive genes and use these ability to filter to focus on the things that are specific to our perturbation. Like maybe that's where drug efficacy or drug side effects are going to be more clearly revealed. And so, yeah, so we like to correct for these, and we think simulating the data with generative neural networks is going to be a way to do that. So this paper is actually still being written too. That's the main theme of this talk, is papers being written. This is actually up on GitHub now if you want to play around with it, though, in this generic gene expressions patterns repo. The source code is there, and you can start sort of using the approach yourself. And I think this will be out pretty soon. The paper's in pretty good shape. So I'd expect to see a preprint in the next month or two. With that, I think I have some time for questions. I would love to thank the folks who make this possible. The lab is a wonderful set of people who create an environment where everyone's dedicated to doing open, transparent science in an inclusive way. And I value that so much. And I feel a great privilege to work with these folks. Collaborators, the folks who were collaborators on the work that I talked about, especially at the end. So we've started working with Jim Costello and Ronnie Powers to begin to compare sort of our generic genes and pathways to theirs. And then on the Pseudomonas work on that last part, Deb Hogan and Georgia Dewing. Funding, so our work has been supported by a number of foundations, by the NIH, a couple different NIH institutes. We've also received the first project I talked about, the software HetNet connectivity search, was actually funded by a grant from Pfizer. And with that, I hope I have a little bit of time for questions if anyone wants to ask any. Thank you.
Thank you, Casey. This was really a very interesting talk. I enjoyed it a lot, and I probably want to ask your comments on the batch effect noise that you're getting in multi-omics data, and although it's there, and then it's kind of like a necessary evil, and then you end up having it and you want to remove it. So when you have such 70,000 data sets that you are trying to do, how much of automation do you end up doing for removing those batch effects?
Yeah, so I mean, the answer right now is it's interesting. So we've been living in this world. Actually, we haven't really been trying to remove it. The good news about this type of approach. So the good news about not trying to remove it is if you have a whole bunch of independent data sets, you really probably don't have to remove it. The challenge of not removing it is, if those data sets aren't truly independent, then life gets a little bit more painful. And so one of the things that, like, I think there's on a data set by data set basis, I actually don't think you should try to remove batch effects. However, on like a technical platform basis, I do think it's probably wise to try to remove them. Thinking about, for instance, like poly A versus ribodeplete methods for RNA-seq, essentially filtering to what you're going to actually sequence in an RNA-seq. Did y'all talk about different types of like poly A selection versus rRNA depletion?
No, not so much. I don't think we have covered that part. Yeah.
Okay. Yeah. So if you have a whole bunch of RNA and you're going to sequence it to see what's there, the thing you would if you just sequenced your RNA, you'd get a whole bunch of ribosomal RNA, and you'd spend a ton of money sequencing ribosomes. And that's not terribly cost efficient. So what people usually will do is they'll either try to use a material that will bind the ribosomal RNA and let other RNA pass through, and then they'll sequence the RNA that passes through, or they'll try to grab onto mRNAs have poly A tails, so they'll try to grab onto the mRNA from the poly A tail. And so because these two different selection methods exist in the data, I mean, there's also a whole bunch of other ones, but those are the main ones. Because these selection methods exist, I actually think you should correct for those because they're pervasive and across experiments. We haven't done that yet, but I think it's absolutely the right thing to do and would make these analyses better. The other thing that I think might be happening, so I have two explanations for this blotch here, right? Things that are generically differentially expressed with the RNA-seq data, but not generically differentially expressed in the microarray data. One potential explanation of these are genes that are not well measured on microarrays, but they are generic, right? So like for some reason, the probe design is bad, there's some reason that they're not actually that well measured. So you couldn't detect them in the first place because they weren't actually well measured on that platform. The second potential explanation is that these are genes that differentiate ribodepletion versus poly A selection. And so they're actually only variable in the RNA-seq data and not variable in the other data. And so, being able to correct for that type of platform effect may help you there. It's something we're working on a little bit. It's not terribly trivial to do, unfortunately. But so long story short, I think correcting for data set is probably not helpful. And so we haven't done that. So ours is very automated in that we actually don't correct for anything. But I do think layering on this next step where you do correct for these large pervasive artifacts is going to make life better. That was a very complicated answer to a very simple question. I'm sorry.
No, actually, it was not the question itself was like quite complicated. So I appreciate you answering that. And also, I think it's quite distinctly contrast. Like you have the microarray versus the RNA-seq results in the previous slide and this slide, that the clarity in which you find the differentially expressed genes is drastically different between these two data. And
yeah.
And it's kind of like I'm wondering if you have any plans to implement this for, let's say, single cell diseased and healthy single cell data, which would be more high quality probably, or it would probably complicate it much further. I'm not sure. So
yeah, that's interesting. So I could see it going a couple of ways with this. So yeah, so you could imagine, like, let's say you do your single cell experiment and then you sort of pull cell types from that. What are the genes that are likely sort of generic in the sense that they're different between cell types, but it doesn't matter which cell types you compare, they're likely to be different. Yeah, we haven't done that. I think the technique that we're using here is essentially a variational autoencoder. I think if you were going to do it for the single cell data, there's another a generative variational method called SCVI, single cell variational inference. So I think I would do the same experiment, but I would do it in the context of SCVI. It's interesting. We haven't done it. We haven't but I am interested in that. The difference in resolution here, like the microarray comparison
Right.
versus the microarray RNA-seq comparison, with the RNA-seq one looking actually better, even though they're different platforms. I think this comes from sample size. So this is, I think, 1,000 samples, maybe even 600 samples, and this is just 70,000 samples, and so I think it's just the sheer quantity of data is helping. But yeah, we could, we haven't done that experiment, but we could subsample this and see if it ends up looking like this.
Right. Yeah. So I have one more question, but then I'll let one participant here ask you that. Suma ma'am.
Yes, sir. Thank you.
We can hear you very feebly. Can you come closer to the microphone?
Yeah, sure, sir. Yeah. So hello, Dr. Cassie. So it was a wonderful session on exploring publicly available gene expression data. Thank you so much for that. So I would like to get more information on, you mentioned about simulated data, right? Yeah, I would like to get more information on that, how that simulation data is obtained for the gene expression, and how much it is able to capture the heterogeneity of biological samples.
Yeah, so that's a great question. Let's see if I didn't put the specific method in here. Yeah, so the way this method works, let's see, where's my VAE? Okay, so we've got an encoder neural network, and then this decoder neural network, and this is actually a latent space that has a defined structure, so you can resample from that. So what we do is we take a data set, a target data set of interest, we embed that into the latent space, so we run this encoder neural network over it, which is this step. And then there's a slide later that kind of shows just a little bit of what we do, although it's in principal component space. So unfortunately, this is in principal component space, and this is sort of, so we're basically taking this nonlinear thing and showing it in a linear space, so it's probably not ideal. But so one of these, I think this is the original experiment. We can then, so this is showing in the principal components of the latent space, you can then randomly move this experiment. And we actually translate it such that the experiment is centered on another experiment. So like you just pick a random experiment. We've translated the experiment to that other experiment. And then you run the decoder neural network after the translation step. So then we're basically running just this network to give you your simulated data. And so what that lets you do is have a template experiment. So I think in this one, this is the template experiment. So we took that template experiment, we embedded it in the latent space, we shifted it in that latent space by translation to the sort of to the centroid of another experiment. And then we run the decoder, and that gives us the simulated data. And so that preserves the structure, but it doesn't preserve the specific genes that are changed.
Okay, thank you. I have one more question. Based on gene expression data, what are the different features you consider for machine learning approaches?
Let's see, can you ask that again? Sorry.
Different features that can be considered for machine learning based on the gene expression data.
Yeah. Yeah, so we have been, so the work, let's see, did I talk about anything here that uses, okay, so I talked, yeah, so for the work that I presented here, the features that we have been using are essentially gene expression values for each gene. And that's pretty much it. But there's a lot of other information you could have. So, this came up earlier, but like one of the features we would love to have would be like our RNA depletion versus poly A selection, right? And you could jointly model that together. I think that would improve these models, and is something we're thinking about. There's a group at UCSC, University of California, Santa Cruz, that has done at least a little bit of work in this regard, trying to kind of automatically detect those. And yeah, I think there's a lot of additional features that can be put into the model if we could get them comprehensively enough across public data. I think if we could do a better job of annotating our data, we wouldn't need to sort of ignore a lot of these effects, like the tissue type, the prep method. And and so I think I'm hopeful that we can continue to develop incentives to lead people to better annotate their data. So then we'll have a much richer feature set, other than just sort of treating the data as unlabeled gene expression values. And then I'm hopeful that we can also develop approaches that go back and re-annotate the old data so that we kind of model it together in a rich way. But yeah, I mean, like all the stuff you would want to do, like really want to do from a machine learning point of view, like, you know, doing some nice, like rich multitask learning, you know, requires sort of better features than I feel like we have right now. There was a really interesting preprint from Russ Altman's lab that looked at, they looked at predicting genetic sex of samples from gene expression data, simply using elastic net logistic regression on Y chromosome expressed and X inactivation genes. And they did a really nice job of showing that, you know, in certain fields, drug trials are more often carried out on male mice, and in other cases on female mice. And, you know, so there's some real, like, I think there's a lot of places you can go if we could get these rich annotations that we could use as features. But it does feel to me like we're in the early days and there's just a lot to fill in. But yeah, I love the question. I think, oh man, if we had really rich feature annotations, the science we could do would be so exciting.
Yeah, that's always I think a dream thing for anyone who's going for feature selection, to pick the right features and then go about it. Yeah.
I know. Yeah.
Anyone else has any questions?
Yes. Thank you first of all for the very nice talk. I have a couple of questions. The first one coming from the variational autoencoders that you were talking about, Casey.
Yeah.
When we say that there are so many technical difficulties or technical issues when it comes to microarray as well as to the RNA-seq. So
Yeah.
how you input your RNA experiments, means gene expression data sets, into the variational autoencoders, because for each, let's say, experiment, you might be having different number of genes and also a different number of samples.
Yes. Yeah. So we are doing it on a sample wise basis. You do have issues with zeros. Let's see. So we've done this a bunch of different ways. I would have to go back and look at exactly what Alex did here to give you a comprehensive answer on what she used in this case. What we will often do is filter to genes that have the highest variability in the data, like often the like 8,000 genes that sort of vary the most, which is, you know, roughly half-ish of the protein coding genes that you could detect. Actually, now that I'm thinking about it, there's at least 17,000 genes here, so she did not do that. Yeah, there's 17,000 genes because there's 17,000. The intersection with this data set, so this is just looking at the intersection of these two, is at least 17,000 genes. So this must include every protein coding gene. I'd have to look back precisely at what we did here, but yeah, the other challenge is, of course, modeling counts. And so there's a few different strategies to use that. In this case, we didn't do anything fancy, which is why I was initially thinking these were the most abundant genes. I would have to, that's a good question. We've done it so many different ways. I would actually have to go back to the method section and see which approach we used here.
Oh, no worries. So the second question comes where you have talked about there are some genetic pathways that get perturbed. It doesn't matter which experiment you are taking. So can we take these generic pathways as a base and use some other kind of learning that comes from the federated learning or something like that to improve the our parent model? So you train it on each experiment, you get some generic pathways and some unique pathways, and then if the pathways are generic across many experiments, then they would be updating the parent model something that the Google also uses when it comes to federated learning?
Yeah, that's really interesting. So the idea would be like, what you do is when you deposit your gene expression data, you also deposit your differential expression statistics. And like, as we look across your differential expression statistics, we want to know, oh, wow, that came up, that came up 15 times out of 20 in the liver or something. That's kind of what you're thinking. And then if it did, then you can sort of include that as a prior. And so you almost expect those to be different. And so you could have, you could think of it as sort of a way of like boosting your power with that as a prior, I'm guessing is what you'd imagine.
Yeah, that was something related to what I had in mind. Like when you have the same pathways coming up again, then do you consider them as like a basal threshold and anything unique or anything different from those unique generalized same pathways coming up would give us insight into like new targets for rare diseases, or is this something too far-fetched to think about?
Yeah. So the way I've been thinking about them, I think is a little bit different than that, but it's an yeah. So the way that I've been thinking about them is like, if I'm looking at the effects of a drug, I would, and like, early stage drug development, I would really love to know what is unique about that drug, not just that cells are perturbed. But now that you've asked this question, I'm kind of thinking like, we have not looked at this, but like, side effects are also kind of generic, right? I mean, like some side effects occur for many, I have the cider example in here earlier on one of those network diagrams. And there's some side effects that occur for a lot of drugs. Right. And so I almost wonder if like you could use the generic pathways as just sort of a way to get it like, like the way I think of these is like kind of almost like, kicking a ball, right? Like how hard did you kick the ball? Like if you see this sort of shift in the generic pathways, this is this size perturbation of sort of base biology, and maybe that tells you actually something about the sort of tolerable concentration of that drug. So like maybe there's information in the generic pathways by how many of the generic pathways are moved and how strongly they're moved versus the specific ones. And maybe, a good drug is one where the specific pathways move a large amount relative to the generic pathways or something. This is me thinking on the fly, and we have not looked at this and we haven't explored it at all, but yeah, those are some really interesting questions. I have no answers for them, but they have raised a lot of questions for me.
Okay. Not to perturb you, but then it was just something on top of my head.
Yeah.
Okay. Anyone else has any questions? Feel free to ask. I'm pretty sure Casey is more than happy to answer your questions.
Okay.
Okay, looks like we don't have much questions from the participants. Probably they are still digesting whatever the talk was. We would probably it will take some time for them to get back to you. Is that okay that participants can connect with you later on, like next 2 days, or maybe after the workshop is over, and then ask questions?
Yeah, that would be perfect. I would love to hear from you. I think these questions have been really wonderful. They've like definitely taken my thinking in a different direction, because I was thinking about these generic pathways as a thing to avoid. But no, I think there could really be something there of going after them as like having signal in and of themselves. So yes, please, definitely. Yeah, questions are wonderful. They're how we think. If you have specific questions, feel free to drop into the GitHub repositories that I talked about and ask them in the GitHub repositories, or feel free to email me or tweet at me or whatever is the easiest way to get in touch.
Okay, thank you for that. So I think, yeah, we'll have one last question. Go ahead.
Yeah, sorry. So just one thought, Casey, that I was thinking. So let's say when we are talking about the generic pathways, and we see those generic pathways in let's say breast cancer. Okay,
Mm.
now we can also obtain the generic pathways that are the pathways that are shifted when a response is given, and when a patient is cured, and versus the patient when it is not cured. And also we have the generic pathways of the disease. So can it be helpful in some way to determine, yes, these are the pathways that should be, regulated in a certain manner so that a drug, can be as effective to cure a patient?
Yeah. Yeah, I mean, I think this is, I don't know the answer to that question. We have just like literally this technology to figure out what the generic pathways are in a setting is so hot off the presses. There's not a printed version of the paper yet. But I think it's just been so hard to go after these in the past because you've had to curate so much data, right? You've had to like curate 100 experiments to be able to figure out which pathways are generic. And so one of the things I think you're asking is like, can you look within breast cancer and say, what are the generic pathways within this context? And to my knowledge, that has not been done yet. We have not done it yet, but it is the type of thing that we can start to ask now. So I guess, I wish I had more answers, but like all science, I guess there's that quote from Vannevar Bush, right? We're going out into the unknown hinterland, and science is fractal, right? There are questions upon questions, and I feel like you were asking the questions where our mind is, but I don't yet have answers.
Yeah, something to explore further, I guess. Yeah. Okay. Okay. I think we have just finished with all the questions, and anyone has any more questions, as Casey said, feel free to contact him after this workshop or during the workshop in the next couple of days, and you can take it from there. Okay. So with this, I would really like to thank Professor Green for giving a very enlightening talk. This was really it ended up culminating whatever topics we had been talking the last couple of days in a compartment wise, and your talk really captured everything in a very holistic way, like networks, gene expression, the next generation sequencing data, and all those things. So I really thank taking your time. I know it's pretty late for you. Okay, so thank you again for coming here and giving the talk. And we look forward to having more interactions with you. I will, thank you for giving your contact details here. I will definitely pass it along to the participants here. That's there in the chat window. So we'll break for a short duration now for half an hour. We'll come back at 11 o'clock and we'll have the second session. And we'll keep you posted in the WhatsApp group about which system to be connected for the next hands-on session. Thank you, Professor Green, and thank you for the talk. Thank you.
Thank you for having me. Bye-bye from yesterday.
Bye. Bye.